Okay, so let's start the class now. So I'll be introducing you the basic concepts of cell-to-cell communication and the pathways involved in signal transduction. And I'll be mostly focuses in my later on classes from tomorrow on more on endocrine system and how the hormones affect cellular physiology and pathology. So today class will be more on the basic introduction on cell biology and how signal transduction affect the interaction between organism and its environment. So to start with, so what is cell-to-cell communication? So cell-to-cell communication is an essential for both multicellular and unicellular organisms. So it is a way in which an organism, whether it's a bacteria or a yeast or a human, interact with each other and also with its immediate environment. It is important because the cell cannot exist by itself. It has to take nutrient, it has to reproduce, and these processes require interaction with the environment. So it is basically through either physical contact or through chemical signaling. So the best example is when you are under threat. So it's the fight response, which is generally people learn about it, that when you are under stress you run or you start fighting. So these are something which are induced by chemical signaling. In higher organisms these are mediated by hormones, but in lower organisms there are also chemical signalings, not exactly hormones, but they do the same thing. They affect how an organism is responding to its immediate environment. So that's why cellular communication is quite essential for sustainability of organisms. So microbes which which existed millions of years back have shown glimpse of how cell signaling and signal transduction happens. For example, I'll touch upon this example of sexual reproduction in yeast. So in yeast, what happens is generally reproduces asexually by budding, but there are also ways in which yeast can sexually reproduce. So the way in which it does so is through the exchange of mating factors. So what happens is that a diploid yeast through meiosis divides into two haploid yeasts. Now they can be they have a different structures, means they can be of A type or alpha. So it's like in higher organisms it's considered as male and female. In lower organisms it's not like that. It's just that these two yeast haploid yeast are of a different constituent and they can reproduce sexually. So how this is done is through the release of chemical messengers. So these are called factors. Like if a type yeast produce a factor, it's called A factor, and there are receptors to receive these factors on a different type of yeast, which is the alpha type yeast. Now what happens is that these two factors are mutually go vice versa between two yeast strains, and when they sense opposite factor, they protrude their cytoskeleton to reach towards the different yeast. So this process is called shmoo. So shmoo is actually the protrusion which happens in the yeast to get in contact with another yeast. So this is how the sexual reproduction in yeast happen, and this is the first glimpse of how two organisms interact with each other by producing chemical signaling. Now in multicellular organisms this goes to a level much more higher, and there are well worked out signaling transduction pathways which happen which help in the interaction of the cells. Now animal and plant cells have cell junction that directly contact the cytoplasm of adjacent cells, and in local signaling animal cell can communicate by direct contact or cell-cell recognition. So for communication between two type of cells, they can either contact through a physical convolution of the two membranes. So you don't have to be apart. They come together just like the yeast cells do, and they form structures known as gap junctions in animals or plasma desmata in plants. So what happens that when these two cells combine with each other through a physical contact, the cellular material can go vice versa between two cells. And also apart from this, you don't have to dissolve the membrane and combine and fuse together, they can also come in contact through receptor-mediated pathway, in which you have a ligand here on the surface of one cell and it has a receptor on other cells, and the cell can signaling the other cell through signal transduction pathway. So either you have a fusion of the membrane, or even without a fusion of the membrane, but you can come physically close enough to transduce a signal to the other cell. So apart from physical contact, much of the signaling in multicellular organism take place through some local regulators, which can be growth factors and hormones, which is generally what we are going to study in our slides later on. So signaling through a short distance is generally known as paracrine signaling. So suppose the cells in the immediate vicinity wants to convey to the other cells in the area that you have to secrete some or you have to affect gene expression in a way which can affect the tissue physiology. So what it does, it secretes growth factors or hormones, growth factors basically like cytokines, and they are secreted and received by the cells at the adjacent area. So these are generally short distance signaling, and this is called as paracrine signaling. Now a type of paracrine signaling also happens in neurons, which is called synaptic signaling. So what happens is this, that a neuron in response to electrical stimulus secrete chemical transmitters known as neurotransmitters, and these are conceived by other neurons, and in this way there's a signal relay which happen during signal transduction in neurons. Now what about the long distance signaling? So when the cells are quite far apart from each other and they have to tell the other cells to respond in a specific way, they secrete their chemical messengers indirectly into the bloodstream. So this type of signaling is known as endocrine signaling, and the chemical mediators are known as hormones. So suppose this is a cell which wants to tell another cells quite far in the body to respond in a specific way, so it will secrete the hormones directly into the bloodstream, and then the blood will take these chemical messenger or hormone to the target cell, and it will receive the signal and respond in a specific manner. So this is how hormones act, and this is called endocrine signaling. So it's generally a long distance signaling in the body. Now all these, whether it's a short distance signaling through paracrine signaling or through endocrine hormones, it follows a specific way in which the signal is relayed in the organism or the cells. So it was first described by Earl Sutherland when he described how the hormone epinephrine act on the cells. So Sutherland suggested there are 3 steps in signal transduction and how the cell communicate with the other cell. So the first step is the reception, is when target cell receive a chemical signaling from the effector cells. The second step is transduction, is how the signaling cascade happens, and last is the response, which is observed in the cell in response to those signaling. So first, let's come to how these different 3 steps happen. So the first reception is on a target cell membrane. You have a protein receptor which will bind to a signaling molecule. Now this step is called reception. So this can be either on the plasma membrane, the receptor, or inside the nucleus. So once this signaling molecules bind to the receptor, then the second step of signal transduction happens, is called the transduction step. This is the relay molecule in the signal transduction pathway. So this change in the structure of this receptor in response to ligand will ultimately relay activation cascade of different proteins, which will transduce the signal from the surface inside the cells, and finally it will affect either the activity of an enzyme or it will have nuclear actions, and this process is called response. So finally, how the cell responds to the stimulus. So how the reception is achieved? So the binding between the signaling molecules and receptor is highly specific. So it is generally not a non-specific signaling. it is a very specific receptor and ligand interaction, which gives a very specific response in the cell. So it is the once the ligand bind to the receptor, it is associated with a change in the structure of the receptor, and this change in the structure helps in the relay of the signaling downstream of the plasma membrane. So most of the water soluble signal molecules bind to specific site in the receptor that bind the plasma membrane. So there are generally three types of membrane receptor. The first is the G-coupled protein receptors. Second are the receptor tyrosine kinases, and the third are the ion channel receptors. So these are all membrane receptors for signal transduction. So the G-coupled receptors are the largest family of cell surface receptor, and they are also known as serpentine receptor because they span the plasma membrane in a 7 helix structure, and they so the 7 transmembrane helix structure, and they act as an on-off switch. So I'll explain you how it happens. So what happens is that as soon as so this is the inactive state of the receptor and its effector enzymes. So when there is no ligand, the G protein is inactive and is bound by GDP, guanylyl diphosphate. As soon as the ligand bind to this G-coupled receptor, there's a switch between the binding of GTP. So GDP is replaced by a GTP where the action it involves the release of GDP from the docking site, and it's activated by GDP. Now, as soon as the GDP is bound to this G protein, it then goes and attached to a enzyme. In this case, is known as adenylyl cyclase, and this conveys the signal transduction downstream. After some time, when the ligand is no more there, this complex is released. So it again goes to the default inactive state, and a phosphate is released from the GTP. So GTP is broken down into GDP and a phosphate. So this is how a G-coupled receptor works. Now the second class of membrane receptor, the tyrosine kinase receptors, actually receptor tyrosine kinase can trigger multiple signal pathway at once, and they generally have a tyrosine which is phosphorylated, and to these phosphorylated tyrosine other enzymes or kinases bind, and they themselves get activated. So this is how this model works. So when there is no ligand, they exist as monomeric forms, this tyrosine kinases, and as soon as there is a receptor, they dimerize and they autophosphorylate. So the enzyme itself has the ability to phosphorylate all its tyrosine moieties. So once there is autophosphorylation happens, it then allows the binding of other relay proteins or kinases to its tyrosine, and these in turns are again phosphorylated, and they execute different cellular response. So the third category now is the ligand-gated ion channel receptor. So these are receptors in which they open or close in response to ions. They open and close response to ligand, which help in the passing of ions inside the cells. So when a signal molecule binds to the ligand to the receptor, the gate allows specific ions such as sodium and calcium through the channel in receptor. So these are generally very much seen in the neurons, where signaling molecules like acetylcholine and adrenergic signaling helps in the opening or closing of these ion channels. So suppose this is a signaling molecule which bind to this ligand-gated ion receptor. It helps in the conformation change which causes this gate to open, and the ions go inside the cell and convey a cellular response. And as soon as the ligand is detached, this gate of this ion channel is no more open, and you lose the effect of change in the ion concentration inside the cell. So beside these plasma membrane receptors, there are also a class of receptor known as intracellular receptors. So when the ligand is hydrophilic, they generally bind to the plasma membrane receptor because it's the hydrophilic environment outside the cell. But when the ligand is hydrophobic, they generally bind to the receptor inside the cells, and they mostly are proteins known as nuclear receptors. So small hydrophobic chemical messengers, they can readily cross the membrane and activate receptor. The examples of hydrophobic messengers are the steroid and thyroid hormone of animals. So they are generally, they are of the family of the steroid receptors. And this activated hormone receptor complex can in turn act as a transcription factor by turning on specific genes. So this is how it works. So in this case, the example of hormone testosterone, it goes inside the cell because it's hydrophobic. It can pass through the membrane, and then it binds to a testosterone receptor. Okay, so these are proteins which belong to the family of nuclear receptor. So they have on one hand a binding domain for the ligand, and on the other hand, they also have a binding domain to bind to the DNA and affect gene expression. So as soon as the ligand bind to the hormone receptor complex, the hormone receptor complex go inside the cell and bind to the DNA, which causes the transcription of the gene, mRNA synthesis, and a new protein synthesis, which affect the cellular physiology. So this is how the hydrophobic ligands transduce the signal. Now, this was all about resection. What about transduction pathways? So you can receive the signal either at the plasma membrane or at the nuclear level. But how the signaling cascade is executed? So signal transduction usually involve multiple steps. So multiple step can amplify a signal in response in few molecules to produce a large cellular response. And this process is known as signal amplification. So the whole idea of signal transduction in the cells is to have an amplified signaling. You can have a single molecule as a ligand, but when it binds to the receptor, it can somehow amplify the signaling, which can affect the whole physiology of the cell. And this is executed by a relay signaling through the molecules, which either have a properties of autophosphorylation and can also interact with a number of different proteins. So you have a one signal, but the response is quite amplified, and this is called signal amplification. So multi-step pathway provide more opportunity for coordination and regulation of cellular response. The molecules that relay a signal from receptor response are mostly proteins. So like falling dominoes, the receptor activates another protein which activates another, and so on. This cascade proceeds. At each steps, the signal can transduce into a different form, usually a shape change in the protein. So this is what signal amplification means. So you have a signaling molecule, the receptor, it activates protein kinase 1. Suppose, for example, a protein kinase 1. Now this protein kinase 1 in turn activate protein kinase 3. So over this process, there is a phosphorylation cascade in which different kinases are involved. Now the beauty of this thing is that the protein kinase 1 can also activate many different kinases. So this is a linear example of how the signaling is happening. But in the cell, this could be a number of kinases which can be activated by the first kinase, and so you get a very amplified response to a single molecule binding to receptor, and this is called signal amplification. It is generally mediated by post-translational changes. So either the protein is phosphorylated or it is acetylated. So there are many ways in which protein can be activated, but mostly the process involves phosphorylation of a specific protein. So small molecules and ions as secondary messengers. So what is this process in which different proteins activate each other? So this is relayed by small messengers, secondary messengers, which can be which are generally not proteins. They can be lipids, and these could be nucleotides. And the best example is AMP. So cyclic AMP is one of the most best studied example of secondary messengers. So they participate in G-coupled receptor signaling. And another important secondary messengers are the ions, and especially calcium ions. So how cyclic AMP works? So cyclic AMP activates is formed by activity of an enzyme known as adenyl cyclase. So if you remember in the G coupled receptor signaling, so once the G protein binds, is activated by GTP, it binds to a protein known as adenylyl cyclase. So adenylyl cyclase converts ATP to cyclic AMP in response to extracellular signaling. Now this signaling, this cyclic AMP in turn binds to a protein known as protein kinase A and causes its conformation change to activate its phosphorylation. So once this protein kinase A is activated, it in turn phosphorylate a number of different substrate enzymes, and that's how the this signaling is relayed by the G coupled receptors. So this is how it happens. So the first messenger is generally the ligand, the hormone or a cytokine, anything. It binds to the receptor. It causes G protein in the active form to bind to adenylyl cyclase, which in turn forms cyclic AMP from ATP. Now this cyclic AMP activates this kinase known as protein kinase A, and this in turn phosphorylates different kinases or transcription factor to mediate cellular response. So how the calcium ion works as a as a secondary messenger. Now why calcium has been acting as a important secondary messenger in the cell is because of its concentration gradient. So generally calcium inside the cell is stored in cellular organelles known as ER and endoplasmic reticulum. So the concentration is quite high in these organelles and also outside the cells. So whenever there is a slight release of calcium from these organelles into cytosol, they trigger a cascade of signal transduction. So this calcium can come from outside the cell through the calcium pumps, which can be activated by specific ligands, or they can also come from the intracellular organelles like mitochondrial ER, which is activated by a through either G coupled receptor or tyrosine kinase receptors. So the way this happens is that so this is a G coupled receptor. It binds to a primary messenger G protein. Instead of going to the adenylyl cyclase pathway, it also activates lipase. The lipase are basically enzymes which acts on lipid. So generally they modify lipid moieties. So in this case, it instead of binding to adenylyl cyclase, it goes to the phospholipase C pathway. Now this enzyme phospholipase C breaks the lipase generally break the bond in the lipids. So they break the PIP2 lipid in the plasma membrane into DAC and IP3. Now this inositol triphosphate, which is IP3, act as a secondary messenger and bind to the IP3 docking site in a calcium channel of a ER. So you can see how these two type of receptors are acting. So one is the G coupled receptor, which was the initial transducer, and then you also have ion gated channels, which is I told you another type of receptors which mediated action. So it's the interplay of these two type of receptor. So finally the IP3 gated channels open and the calcium comes outside from the ER to the cytosol. And as soon as the calcium comes outside, it goes to and bind to a specific class of kinases known as calcium activated kinases like CAM kinase, which is calcium activated kinases, and these in turn phosphorylate and activate other downstream proteins. And this is how the signal transduction in response to calcium happens. So now the third step in cell signaling is the response. So I've told you reception, transduction. So next is response. So after a signal is transduced, how the cell responds to it. So this is basically in terms of either cytosolic response or a nuclear response. So the response may occur in the cytosol. Maybe it changes the activity of enzymes which are affecting the metabolic status of the cells, or it can affect the gene transcription. So this is the response in which the cell behave in response to a outside signaling. So for this this example shows you how in response to growth factors there is a change in gene transcription. So the growth factors binding to the receptor activate a phosphorylation cascade, which in turn activates a specific transcription factor. So this is the response, this is the transduction, and this is the reception step. So as soon as the activated transcription factor binds to the DNA, it initiates the gene expression changes in the cell. Other pathways apart from nuclear action is through the activity of enzyme, as I've mentioned. So they can be change in the enzyme activity as such. So the difference is when you have a change in response to a stimuli in terms of cytosolic response, it is generally very fast. So and it is considered non-genomic activation. So it acts very fast, and within minutes you can see the effect in terms of enzyme activity. But when a signal to a cell is meant to affect the nuclear response, it takes some hours to happen, even days. So the reason is that for a signal to affect the gene expression, it has to first go to the cytosolic step of activation, then affect the nuclear receptor. Nuclear receptor in turn takes some time to transcribe a specific genes. Then mRNA will be translated into protein, and finally that protein will affect the cellular physiology. So the nuclear cascade is a bit slower, and the cytosolic response is much more faster. But at the same time, cytosolic action is short-lived also. So it goes and then falls very fast. Whereas the nuclear action is for sustained response of a cell to external milieu. And is generally seen in terms of hormone action, which are steroid family receptor hormones. But in case of epinephrine, when you need fast response, you generally affect the cytosolic response. So it's very fast membrane receptor action. You do the job and then it's finished very fast. So because signal transduction is a very important process in cellular signaling, so there are many ways in it in which it is fine-tuned. So one is the amplification of the signal, as I have told you. The other is the specificity of the response. So you cannot have a non-specific response to a cellular signaling. And overall efficiency of response is enhanced by proteins known as scaffolding proteins. And finally, you need a timely termination of a signal so that you don't overdrive the system or overactivate a cellular response. So signal amplification is something which I have told earlier, is that it amplifies the cellular response, and in each step there are a number of activated products much greater than the preceding step. Now the specificity is something which occurs either because of the receptor being different in different cells. So two different cells can respond in a very different way to one substrate or the ligand. And it can also happen because of the downstream proteins which interact with the receptors. And there could be a crosstalk between two different cellular pathways, which ultimately affect the coordinated incoming signal. So these are the different type of response you can expect in a multicellular organism. So you have a signaling molecule, you have a receptor, you have a relay molecule, and you get response one. So you have a one type of response, either in terms of transcription of a gene or activation of an enzyme. Now these relay molecule can in turn activate two different type of downstream enzymes or kinases. In that case, for a single same signaling molecules, you can have a two type of response going together. So suppose there could be a cell proliferation response, and there could also be a change in the cytoskeleton dynamics in response to a single ligand. But unlike the other cells which only gives one response, this type of cell has a different response which can go parallel, because the relay molecules have a different interaction pattern. Now there are also crosstalks. So the crosstalk is that, suppose you have this ligand which we have discussed in these two panels. So this relay this signaling through the cascade of a protein, but whereas another signal also come, it has a different receptor, and somehow it can activate a kinase, which can in turn affect the activity of this protein, which was a target of the initial signal. Now this could be inhibitory in action, or this could be co-stimulatory in action. So it could be that you have antagonist ligands. So suppose this ligand activates activity of a specific protein, but this ligand is meant to inhibit it. So as soon as the concentration of this ligand increase, this will inhibit the activity of the first substrate signaling. On the other hand, you can also synergize if this pathway corroborate or have a further phosphorylation event on this protein, so that a combined action of these two ligand can even amplify your signal furthermore compared to a single amplification. And this response 5 this exactly is because of different receptors. So if the receptor structure shape like this compared to this, you have a different response, and the relay molecules could be of different types or not. Okay, so this is the diversification in which you can have a signaling communication. Now, efficiency of signaling can also be increased by a specific class of protein known as scaffolding proteins. Now, scaffolding proteins are large relay proteins, which other relay proteins are attacked. So it is like a platform. So just consider it as a receptor trying to phosphorylate 3 different type of kinases after activation. So in randomness, these proteins can come and go and come and go. So that will take time. So what the scaffold protein does is that it bring all those 3 proteins, the target proteins, in a hub type of system, and the scaffold protein in turn connect to the receptor. So what happens that the time to activate the other 3 proteins by the receptor is remarkably decreased. And an example is this. So suppose these 3 different kinases are meant to be activated by this receptor. Upon activation of ligand, the scaffolding proteins will bring all 3 of them close to this. So this scaffolding protein itself is not phosphorylated, but the conformation change which is induced by the action or binding of the scaffolding protein to receptor somehow brings the other 3 protein kinases very close to the receptor, and it helps in either the autophosphorylation or phosphorylation of this receptor mediated action on this on these 3 proteins. So this is how you can amplify the signaling also. And finally, the termination of signaling, because you cannot have a perpetual activation of cellular signaling because it may be pathological in nature if it goes beyond some time. So you have different way in which the signal is terminated. So if the ligand concentration fall, the fear receptor will be bound. So one way is that the ligand itself goes down with time, and unbound receptor reverts to inactive state. The other way is that you decrease the concentration of the secondary messengers. So for example, cyclic AMP is degraded to AMP by action of an enzyme known as phosphodiesterase. So because cyclic AMP is the major mediator of the signaling cascade, the best way to terminate the signal is to terminate the action of the secondary messenger. So this is one way of stopping the signaling cascade. Now the other way in which you can stop the signaling cascade is through dephosphorylation of a phosphorylated kinase or protein. So there are phosphatases, just like there are kinases which phosphorylate the target protein. There are also phosphatases which remove the phosphate bond from the proteins and in turn inactivate them. So this is a process in which you can stop the signaling relay from a signal transduction pathway. So this is for the first lecture. Thanks. And if you have any question, just let me know. So tomorrow we'll focus more on the endocrine aspect. So any questions?